In 2012, Shinya Yamanaka was awarded the Nobel Prize in Medicine for demonstrating that an adult cell can be reprogrammed back into a pluripotent state. In other words, it can be transformed into virtually any cell type in the human body. That discovery reshaped modern biology. Today, that science has become an approved therapy. Japan has authorized the commercialization of the world's first treatment based on induced pluripotent stem cells, iPS cells, for Parkinson's disease. This is not an experimental trial. It is an authorized product for manufacturing and sale. Parkinson's disease is a neurodegenerative disorder affecting hundreds of thousands of people. In Japan alone, more than 250,000 patients are estimated to live with the condition. Until now, available treatments have only managed symptoms. They have not restored the neurons that were lost. The key lies in dopamine. In the brain, dopaminergic neurons produce this essential neurotransmitter responsible for controlling movement. When these neurons disappear, tremor, rigidity, and progressive functional decline begin. Starting from human iPS cells, researchers generate specialized dopaminergic neurons in the laboratory. These cells are not maintained in isolation. They organize into three-dimensional structures known as dopaminergic neurospheres. Each neurosphere contains thousands of functional neuronal cells. They are structured aggregates capable of maintaining neuronal identity and producing dopamine. The images illustrate the process. iPS cells in culture, differentiation into neurons, and the formation of these complex cellular spheres. The next step is transplantation. Dopaminergic neurospheres are implanted into the patient's brain, in regions where neuronal loss has been most severe. The objective is not simply to introduce cells, but to restore biological function, to recover dopamine production and reactivate neural circuits. In magnified representation, one of these neurospheres appears as a central core from which neuronal projections radiate in all directions. Each sphere can contain approximately 10,000 differentiated dopaminergic neurons. This is not a passive structure. It is organized, living tissue. These cells do not merely survive cryopreservation. They retain their functionality and their ability to produce dopamine after transplantation. Japan's approval marks the transition from research to clinical reality. Science initiated by a Nobel Prize has now become treatment for real patients. And with it begins a new stage in regenerative medicine, the transition from basic research to the clinical and commercial production of advanced cell therapies. Japan takes the first step. The science of iPS cells, initiated by a Nobel laureate, has now become a real treatment for real patients. And with it begins a new era in the functional restoration of the human brain. The technology used to achieve this milestone is proton magnetic freezing, which marks the start of a new era in the cryopreservation of biological products, leaving nitrogen behind.